Hi, everyone. Welcome to OsteoBites. Thank you so much for joining us today. My name is Christina Iptoma, and I'm the Director of Scientific Programs at MIB Agents, and also mom to OsteoAngel Dylan. Today on OsteoBites, we are joined by Dr. Alex Davies from Oregon Health and Sciences University to discuss his research on dynamic tumor microenvironment signaling crosstalk and signal integration using live cell microscopy techniques. Thank you so much, Dr. Davies, for joining us on OsteoBites today. We are thrilled to have you. And thank you also to our panelist, Walker, for joining us. Please feel free to submit questions during the presentation through the Q&A in Zoom, and we will share those with Dr. Davies after his presentation. A little bit more about our guest today. Dr. Alex Davies graduated with a PhD in biochemistry and molecular biology and a DVM with an interest in comparative oncology from the University of California, Davis. He then completed a postdoctoral fellowship in cancer biology at Lawrence Berkeley National Laboratory before joining the Ohio State University as faculty in the Department of Veterinary Biosciences. And while at OSU, he was a member of the Comprehensive Cancer Center and faculty in the cancer biology and cancer engineering programs. Currently, Dr. Davies is an assistant professor at the OHSU Knight Cancer Institute within the Division of Oncological Sciences and Cancer Early Detection Advanced Research Center, also known as CEDAR, where his work focuses on dynamic tumor microenvironment signaling crosstalk, signal integration, and the development of 3D or organotypic and tissue models to study these interactions using live cell microscopy techniques. Thank you so much for joining us today, Dr. Davies. We're really looking forward to your talk. Just a few announcements before we get started. Anyone who regularly attends or listens to OsteoBites already knows that Factor 2025 is June 26th to 28th in Salt Lake City. Also, fresh news coming in, Dr. Davies will be there. So if you're really interested in today's talk, you can come to Factor in Salt Lake City and engage with him directly on this topic. We are also accepting travel award applications now for scientific attendees. The due date for that is March 28th, so you have a little bit of time. We're also offering a limited number of travel awards for osteo warriors by invitation. Social workers, child life practitioners, and medical professionals can nominate osteo warriors between the ages of 8 to 22 who are able to travel to Factor, but would just require financial assistance to do so. The due date for that, for the Osteo Warrior Awards, is a little bit later, sometime in June. So if you know of anyone who might be interested, if of any patients, please for sure think about nominating them. I also wanted to mention, just kind of apropos to our presentation topic today, MIB Agents has established an international working group dedicated to the study of heterogeneity in osteosarcoma by single-cell technologies. And by sharing the knowledge and establishing standards for the collection, analysis, and dissemination of single-cell data in osteosarcoma, the group seeks to advance osteosarcoma single-cell research toward improving the care of osteosarcoma patients. The group has a great acronym. It's called CURE-OS, which stands for the Collaborative Understanding of the Range and Evolution of Osteosarcoma Single Cells. I'm going to put more info in the chat about this group, where you can find more information or join the working group if interested. Walker, could you please introduce yourself?
Yeah, hi, my name's Walker. I've been on the junior advisory board for a few years now. I'm an osteo warrior diagnosed in 2018, and then I've been NAD for about the last 5 years, so very excited for that. I had a chance to kind of take a sneak peek at the slides today, and I have to say I'm really excited to hear more about it, and I'll probably be one of the people bothering Dr. Davies at Factor asking more questions. So you can take it from there, Dr. Davies.
All right, thanks for the introduction and thanks for the invite to present. So I'll go ahead and put my slides up. And as mentioned, if there are any questions during the talk, please post them in the chat. And if they're clarifying, we can stop and address those. I want people to follow along and make sure that everything makes sense as we go. So I'm happy to talk about these live cell models that we're developing to explore questions about heterogeneity and drug response variation in diseases like osteosarcoma. And I'll talk about a story that is a collaborative story and how we're using these models to understand the biology and how to better treat these diseases. And so I want to start out by talking about heterogeneity, because we talk about this a lot in tumor biology and the concept of intratumoral heterogeneity, but there are a lot of different forms, and that can be genetic heterogeneity, fixed heterogeneity. or it can be phenotypic heterogeneity, for example, where cells transit between different states over really short time scales of minutes to hours and days. And so what I'm primarily going to be talking about is this concept of dynamic heterogeneity, where if you have cells that are in a particular state, it's not that they just are fixed in that state. They can actually move between them at any given time. And so this is important for why cells, invade and metastasize. It's also important for why cells may respond to a drug at a given time or not, right? And importantly, it's not a linear process to go from, for example, in this diagram, going from one color, to another. You can switch between these states, right? And we don't have a really great understanding of this process. And so what we do know is that there are multiple factors that can influence this. There could be cell intrinsic processes where cells are changing their transcriptome and proteome. It could be host immune influences or signals in the local microenvironment, or the interventions themselves that all combine to push these state differences, right? And so as we think about why this matters, I mentioned in terms of treatment, I pulled this image of a patient that was subjected to treatment, and you can see that, on these scans, there are some tumors that respond and other tumors that don't, right? And presumably these all came from the same origin, but they have slightly different behaviors, and those could be genetic, they could be phenotypic, or they could be a combination of both, right? And so we're focused more on that phenotypic aspect and really asking the question, how do microenvironments, so where these cells are, whether they're in the liver or the lung, or even within those organs, what they're next to, a blood vessel or an immune cell, how those converge to control the cell states that we see, and how that affects whether a cell responds to a drug in the way we would predict or not. And so for me, my motivation is really like, what could we do if we could see these individual cells respond to their signals in microenvironments? How would that advance our understanding and ability to treat these diseases? And so my lens of looking at this problem is through the use of biosensors and building tissue models, and then using computational methods to analyze this problem. And so that's really what we're going to talk about for metastatic. So what I'll do is I'll start by talking a little bit about these live cell models that we're using and developing to study these processes. And this work couldn't be done without these people. I can come up with ideas all day long, but it takes really talented people to make them reality. And so Rowan has really led this project, along with Bea, that's contributed to developing the models, and Jeremy is an amazing computationalist and biologist. So thankful for these people that have done all the work I'm showing. So to begin, we're really in an age where we can measure these processes because of advancements in biosensors. And so what I'm showing you here is, let's see if I can actually put my pointer on, is some sensors that we use in particular to measure these sort of dynamic changes in cell state. And so if you look here at these signaling reporters, they're constantly expressed in cells, and they move between the nucleus and cytoplasm of the cell. So here's the boundary of the cell, and here's the nucleus. And as the reporters move, we can measure that activity, and it gives us a quantitative readout of whether or not a cell is receiving a signal, and for how long it's responding to that. And this movie that's playing a little fast here is, a cell cycle reporter, so we can tell what state a cell is in terms of its cell cycle, for example. Or we can do these more complex things, which is what I'm going to talk to you about, where this tumor cell is sitting in a lung tissue, and we can track its fate so that it has a signaling reporter, and we can track its position and its response to a drug where it's dying, so you see the loss of fluorescence. So we can combine these types of techniques to really monitor over time what cells are doing in an environment that mimics the host or patient, right? And so the way we've done this is we've developed these models using mouse models primarily. I'm not going to talk about the human work because it's a bit more complex. But the basic idea is that we take reporter cells. So this could be an osteosarcoma or some other like a breast cancer cell line. We have another project in the lab doing breast cancer. But we take these cells that have reporters of interest in them for signaling, cell cycle, and transcriptional reporters to then inject them into mice, and we can then harvest tissues out of them. So what I'm representing here in these cartoons is that the tissues are bearing metastases in the lung or primary tumors in the mammary gland, and then we can either derive organoids or take lung slices. So this is a cross-section of a piece of lung, and so you can see the whole lung field, and these are all individual tumor cells in that lung field. And the cool thing about this is that we can do it at any time point. So what I'm showing you here is really as these cells have just seeded in the lung, so they've just spread from the tumor into the lung tissue is what we're mimicking. And we can take that initial stage, or we can take it much later when you have an established metastasis and it's a bigger nodule, which is what I'll show you on some of the later slides. And so this gives us a lot of flexibility to understand the process, but in the context of this living tissue. So that's the important point to make clear is these are all living, right? So the whole tissue is living, the tumor cells are living, and we can measure those interactions through live cell imaging and then computational analysis model. All right, so this is what this looks like in practice in terms of the workflow, is that for the lungSITE model, we can take an intact lung and then section it into slices and put them into multi-well dishes. And so this is just a snapshot of one well of a dish, and you can see all these individual tumors that we can then profile. And so these multi-well plates are put onto microscopes that have live cell imaging capability. So they have an environmental chamber that controls humidity, temperature, CO2, oxygen, all of it, right, to keep these cells alive. And then we can track these individual points over time for periods of days to weeks even. And so we can take a snapshot of these individual lesions every 20 minutes for that duration. So we get a massive amount of information over time and space. Okay, so this doesn't come without its challenges. So we've had to develop computational pipelines to really get the information out of this, because it's not just an observational science, it's a quantitative science. And so the way we've approached this problem is to use advanced segmentation algorithms, where we essentially have to hand draw and train the models to pick out and figure out what an actual tumor cell looks like, and what its neighbors look like, and also what the tumor microenvironment looks like. So in this case, this is a lung, and you can kind of see the lung tissue in the background here, and these tumor cells living in it, which is maybe a little more apparent in this image here. But the computer has to accurately be able to pick out all of these things and pick them out over time. And so you can see that problem here, where this is a tumor nodule, and each one of these blue borders is an individual tumor cell, and the green outline is actually the nucleus of those individual cells. And so you can see this cell is kind of invading out into the airway. And then over time, the cells go away because they were treated with a drug that kills them, right? And so we can get a measure of when they die and how they die and where they die, right? So you're getting a lot of information. And then along with this, because we have these fluorescent reporters, we can get a readout of the signaling activity in those individual cells and how they're perceiving their environment or responding to the drug with respect to, for example, an ERK signaling pathway or an AKT signaling pathway. And then add on to that, we can then build relationships between what the cell environment looks like. So how many cells are touching themselves that are cancer cells? How are those cancer cells interacting with the normal cell boundary, so the lung itself, the signaling information that correlates that? How does it interact with the ECM boundary, and what percentage of the cell interacts with that? And then we can relate this all back to what that cell's trajectory was, or fate. Did it die? Did it grow? Did it invade? What are those characteristics, right? So then we can go back and just try to understand this process in greater detail. So I'm going to transition into how we're using this as a model to understand osteosarcoma. and really to understand why individual cells respond or don't respond, and to identify new drug combinations in clinical trials.
Dr. Davies, before you launch
Yeah.
into this part, can I just ask a basic question about the previous and the development of the model? So it looks like the model is tail vein injection, right?
It is, yeah.
And so curious just that using that versus like an orthotopic model and how that might affect kind of the lung mets, like the characteristics of the lung mets.
Yeah, absolutely. So in general, you know, sorry, I'll go back to the slide. In general, you know, this is pretty well accepted methodology for, you know, introducing lung metastases for osteosarcoma. However, it's not without its caveats that you're pointing out, because if you have an orthotopic model, for example, there are potentially things like exosomes that are moving around the circulation that are priming the tissue microenvironment, and you may miss those steps in this process. And so we do have some interest. We have not done this, but we do have some interest in working with orthotopic models or spontaneous metastasis models. And I think, you know, some of the models out of MD Anderson that are arising with the mouse models there, and even some of the cell lines from the Sweet Cordero lab with the PDX models represent an opportunity where we could potentially implant in the primary site, wait for spontaneous metastasis. So I think that's a great point. But for everything I'm showing you here, it's going to be the tail vein injection.
Got it. Thank you.
Yeah. All right. So so getting back to this question, sort of the central theme of the work that my lab has been doing is really understanding how signals that arise from the local tumor microenvironment influence signaling in the tumor cells, and how that dictates cell fate. And so the pathways we've primarily been interested in are growth factor signaling pathways and receptor tyrosine kinases. And so I'll show you some reasons why we're interested in those in particular. But some of the key players or effectors that are downstream of receptor tyrosine kinases are these kinases themselves, ERK and AKT, that are part of these signal transduction cascades. And so ERK and AKT regulate the expression of downstream target genes, and there's a whole plethora of genes that they regulate. I'm only going to focus on a really small subset of that. But what's important about this is that these genes regulate cell fate, right? They can regulate the transcriptome directly, and they can regulate drug response. And so in particular, I'm going to talk a lot about this protein FRA1, which is highly regulated by ERK signaling. And it's a great readout of ERK in and of its own right. So it's kind of a surrogate for ERK activity, but it's also a transcription factor and a pioneer factor. So it can go into and essentially it's an AP1 transcription factor. So it's involved in a lot of processes, including EMT and things like that. The other protein I'm going to talk about is MCL1, which is a pro-survival protein that's regulated transcriptionally and post-translationally by ERK and AKT signaling. And so all the signals that come in through these pathways from the environment are going to regulate these downstream processes, right? Okay, so to just give you a little background on that, I'm going to highlight a couple of findings that Ryan Roberts and I published recently, describing the role for the tumor microenvironment in establishing metastases in the lung. And so this study was a great collaborative effort between the two of us, and really paired our labs to expertise. And what we found was that by looking at when metastases are establishing very early in the lung, we got a good picture of what changes occur. And so what we found was that different cells, so this is a transcriptional profiling of the lung tissue, is that if you look at different cells that compose the lung, such as fibroblasts, monocytes, and macrophages and things like that, they all produce different growth factors. But there's this initial sort of boost in growth factor production by the lung itself once a tumor cell lands there. And so, you know, things like fibroblast growth factor and these EGF growth factors kind of peak in the initial phases of colonization. And so what that ends up doing is activating ERK signaling in the local environment, or excuse me, in the tumor cells in the sort of local microenvironment. And you can see that represented here by this marker FRA1, which I mentioned is the downstream target of ERK signaling. And so it follows this temporal sequence where you initially get growth factor expression that corresponds with ERK activity and FRA1 expression over time. So you get an initial peak. Well, what's interesting about this in terms of the, what I was talking about with intratumoral heterogeneity is that you can appreciate some cell-to-cell variation in FRA1 expression. And so what this is telling you is that even though these growth factors are upregulated in the microenvironment, they don't have a uniform effect on every tumor cell, right? So each one of these cells is responding a little bit differently to its microenvironment. And to be fair, there may be some intrinsic things that are different about these individual cells, but we've done some work to show that this is really a big, this is influenced heavily by the microenvironment, that this heterogeneity that you're seeing here. And so with those data in hand, we wanted to understand whether or not there were other downstream pathways that are regulated by ERK that might actually be targetable. So FRA1 is not particularly targetable. We don't know what its specific processes it's regulating in osteosarcoma per se. But what we did was a search of the data that Ryan's lab had generated and the transcriptomic data and asked, are there vulnerabilities or potential vulnerabilities that are regulated by ERK signaling? And it turns out that we identified this protein MCL1 as being highly upregulated. And so MCL1, when it's abundant, favors a pro-survival state in tumor cells and even normal cells. And it's extremely regulated by RTK and ERK signaling. So what happens is if ERK is active, it actually stabilizes the MCL1 protein and promotes this pro-survival state. Whereas if ERK signaling is low, you favor an instability or more pro-death state, right? And so we asked whether this represents a vulnerability in the lung, because we knew that the growth factors in the lung microenvironment were activating ERK, and we knew that MCL1 was up. And so that's represented here at the protein level, where we see a similar relationship that early we get an increase in MCL1 and FRA1 expression that corresponds with ERK signaling and signaling in the microenvironment. And you can see that corresponding increase in MCL1 here along the sort of initial time points and then tailing off. And I should have mentioned that we're comparing this to primary tumors that are in the bone. So this is the relative difference in the lung versus the bone. And so indeed, this does represent a vulnerability. So we performed mouse studies using the OS17 cell line. And what you can see here is that in the vehicle-treated groups, there's a large tumor burden, very large lesions. If we treat with cyclophosphamide as sort of a mimic for ifosfamide, you get a reduction. MCL1 as a single agent it actually reduces the tumor burden significantly. But then when you combine this, we actually saw some mice that didn't have any evidence of metastasis at the time of, at the end point of the study, right? So you're getting some additive effect with these drugs. All right. So coming back to how I framed this question about heterogeneity, I was really struck by the potency of MCL1 inhibitors, but I was also intrigued by the fact that some tumors didn't respond to it fully. And so this gets back to our question about heterogeneity and why some tumor cells respond and others don't. And so, I think our model gives us a really unique window into this to be able to ask at the single cell level within a tumor microenvironment why one cell is responding and one's not, right? And so what we did is a follow-up to that study is we went back and we re-engineered these osteosarcoma cells, the OS17 cell line, to express reporters that we were interested in. So in this case, we have these cells expressing an ERK sensor, an AKT sensor, both as monitors for what these cells are seeing in their environment and the signals they're receiving, a downstream FRA1 sensor. So in other words, what's integration of these signals into a downstream target, FRA1 being a target of ERK, and then a nuclear marker that allows us to actually track these cells computationally. And so this is what this looks like. So this is a 2-week-old osteosarcoma or OS17 nodule within a lung field. And so these are individual cells. You can see the individual nuclei, and you can see them start to signal, which is this movement of the reporter in and out of the nucleus. So you can see the sort of brightness go away, and they start to blink, and that's them actually receiving signal. And then in response to that, they start to produce this FRA1 signal, right? So it allows us to track this sort of flow of information within the lung. And so if you compare then the heterogeneity that we saw with respect to FRA1 expression in the lung of mice, so these were the ones that were sacrificed at the early time points of the study I showed you, you can see that our reporter recapitulates that heterogeneity from cell to cell. So some cells are really bright for FRA1 and others are really dim, right? And so you see that relationship. So we're recapitulating the in vivo in an in vitro setting. All right, so this is what this starts to look like as you get into more developed nodules. And so we can start to pick apart relationships that explain some of this underlying heterogeneity. And so what we found is that if you look at the tumor core, there's relatively low ERK signaling, and you can see the reporter remains primarily in the nucleus. Whereas in these sort of peripheral sections where the cells are kind of exploring their environment in the lung, you can see these little protrusions coming out and the cells are more active. The ERK signaling is typically on, and we see that represented both in the reporter here, the ERK signaling reporter, but also in the expression of FRA1. So what you're seeing is in real time, environmental and geographical regions that promote ERK signaling and FRA1 expression. So sources of heterogeneity that relate to the spatial and temporal positioning of these cells. And so in general, we can start to draw these boundaries where we see these ERK and FRA1 high areas and ERK and FRA1 low areas in the tumor core. So a question that arises from this is whether or not that relates back to that initial relationship of pro-survival pathway upregulation. So I already told you that ERK signaling stabilizes MCL1, and MCL1 is important for the stability of lung nodules, right? So if we use an MCL1 inhibitor alone or in combination with cyclophosphamide, we can eliminate a significant number of tumor mets. So we wanted to ask whether this spatial relationship in terms of signaling and expression of ERK targets, MCL1 being one of them, relates to whether or not these cells die in response to treatment. And so the prediction would be that high ERK signaling that promotes stability of MCL1 would correlate with better individual tumor cell survival versus the ones that have low signal. And this happens to be one case where our prediction was true, not always the case in science. And so what we found was that this relationship sort of happened as we predicted, where the tumor core, where the cells have low ERK signaling, died first, and then these cells on the periphery were dying later. But what was interesting is that we found that there were these persister populations. So instead of just a linear relationship between sort of survival, so death in the core and survival on the periphery, there was this third population where cells initially looked like they were sort of intermediate signaling and, they're not showing FRA1 expression, which you can see in this movie here. But as the cells start dying, then the ERK activity ramps up and the FRA1 expression ramps up. And then these cells actually persist, right? And so I'm going to get into this in a little more detail. So if it didn't 100% make sense, hopefully this next slide will help clarify that. But the basic takeaway is that there are these cells that do persist, and this is clearly important from a clinical perspective in understanding persistence. So what we first noticed is that, as I mentioned, death occurs from in to out. So we noticed that at the tumor core, all these cells started dying, and then these cells out on the periphery either made a survival or death choice. And it may be a little bit easier to see that represented here in the computational data, where we can track individual cells and their boundaries. And so you can see these two cells, something's different about them, that they're surviving while these other neighbors are dying, right? And so we could go back then computationally and plot what their signaling activity is and when they died, right? So what was their history and what was their future response? And what we learned was that, you know, it's actually not as simple as just being ERK low versus ERK high. So over time, we saw an increase in ERK activity, presumably supporting MCL1 stability and other pro-survival factors. And surely, if you look at these time points at the end and the beginning, there is this, you know, low ERK signaling and high ERK signaling. But some of these cells that are dying are already trending up in terms of ERK activity. And we can see that kind of here when you break it down into sort of these arbitrary bins of the initial states, early death, late death versus persisters, is that the early death, it's sort of in between. There's some ERK activity, so you know, it's not as if just turning on ERK is going to promote survival in every cell. There's something that's special about these individual cells down here that are persisting. And this is actually known because ERK actually plays a role in activating cell death as well as promoting cell survival. So it's a very complicated sort of Goldilocks-like phenomenon where it has to be just right to promote one or the other. But we asked why there are these differences here and how we could sort of explain this process in a little greater detail. And it turns out that the spatial relationship is really critical. And this is an important point because this really highlights that it's never just going to be the tumor cell and it's never just going to be the microenvironment. It's the combination of both that matter, right? So as I said, these persisters have high ERK activity. And so then we went back and mapped what was their relationship to other cancer cells in terms of their positioning, and what was their relationship to other lung cells in terms of their positioning. What we found was that these persisters were always in some intermediate proximity to another cancer cell. So they're presumably receiving signals from their other tumor cell kind of, you know, neighbors, but they're also right next to a lung cell. So they're receiving signals and cues from both sort of players in this microenvironment. So on average, they always have a high ERK signaling. They're generally close to other tumor cells and close to lung cells. So it sets up this nice relationship. And so with this data in mind, we got to thinking about what could be processes that are going on that are leading to this this resistance that would coordinately use the host microenvironment and the tumor cells. And so we started to think about it, and it was kind of obvious, right? The tumor cells that are in the core are blowing up. Their membranes are exploding, and they're spewing their contents all over into the neighboring environment, right? And so there are bioactive factors in cells, and they're going to be released and perceived by their neighbors. And so we asked whether or not there were relevant factors that could perhaps drive up ERK signaling that are being released by lyse tumor cells from the drug. So we treated these tumor cells with MCL1 inhibitor, and right at the time when they start to die, we collected all the soluble contents that are being released. And then we performed a cytokine array to detect growth factors that they release. And so what we identified was a whole panel of growth factors, and there are commonalities and differences, and this would require probably a whole talk in and of itself to go through. But what we started to see was that there were patterns, and they weren't just specific to OS17. They were common to other osteosarcoma cell lines as well. And so what was notable about this was that we found a bunch of growth factors that interact with receptor tyrosine kinases, these RTKs, namely EGF receptor, FGF receptor, IGF, and PDGFR. These are all common targets that have been explored in osteosarcoma and are of interest, and so they have some documented relevance. And what's interesting is that we do see common patterns of expression, and some of this has to do with the semi-quantitative nature of this type of assay. But we do see some induction as well as just general expression. So it seems to be there are a couple of things going on where the tumor cells may actually start to upregulate some of these factors when they're starting to die, but they also are producing them already and then spew them into the environment. So with these data in hand, we ask the question of whether or not this actually has an effect, right? So we can measure it, but does it actually do anything? And that's the critical part. And so to test this, we took cell lines, tumor cell lines, and then we did mechanical disruption. So we did mechanical disruption specifically because we didn't want the effect of MCL1 inhibitor to be translated onto our test population. So I'll get into that a little bit more in a second. So we basically broke these cells apart so that all their contents were released, but they didn't have MCL1 inhibitor around or any other drug around that could interfere with our assay. So then we took the contents of those cells and we put them back on the parental cells and asked whether that turned on their signaling. So essentially, we're trying to model whether a dying cell next to a live cell is capable of inducing signal. And so what we found was that that's quite true. So if we just add lysate, you can see with this ERK signaling reporter that if we add lysate, there's a peak in ERK activity and it kind of trails off over time. If we add a MEK inhibitor, which interferes with the ability to activate ERK signaling, it blocks that signal. So it tells us it's happening through that pathway. So then we went upstream and asked, well, which receptor pathway is it actually working through? Because we had identified growth factors from all these different pathways. And what we found was that initial stimulation was very specific to the FGF receptor, arguing that fibroblast growth factors were a key player in this role. And there's some interesting data in here that we see consistently in other studies is that, and I kind of want to note this, even though it's a slight digression, is that treating with different inhibitors may actually give you different responses than you think. So not only did PDGF receptor inhibitor not block this induction of ERK activity, it actually enhanced the induction of it. So you can see a higher magnitude, higher amplitude response, right? So you have to think very carefully and test very carefully these combinations, because these receptors, while they receive information from different growth factors, the receptors themselves and the downstream regulatory elements talk to each other. And if you inhibit one, you can affect the other. You have to be very careful and thoughtful about the way that we do these things. Okay, so with those data in hand, we went back and we tested this a little bit more specifically and asked, does this really truly correlate with when the cells die in terms of being able to induce that ERK signaling? And so we used those same cell populations with an ERK signaling reporter, and we treated them with MCL1 inhibitor right here at time 0. And what we see is that as they start to begin to die around 4 hours, we see a peak in ERK activity that kind of corresponds with what we saw in that lung. And if we treat with, so here's MCL1 inhibitor alone, and we see a peak in ERK activity that lasts during the cell death period. And if we block it with MEK inhibitor, we see nothing. If we block with FGFR inhibitor, we see nothing. So it recapitulates what I just showed you, where the cells are actually dying and just doing this themselves. And so we then ask the converse situation that if you just add the growth factor and treat with MCL1, does that promote survival? And indeed it does. So these experiments are showing you that the inhibitor is inducing lysis. Lysis is inducing signaling because of the release of this growth factor. And if we artificially add more of this growth factor around, we can actually promote survival in the face of this inhibitor. So it really kind of is putting more nails into this this story of the growth factor promoting survival. And indeed, if we do this experiment, but then we add a receptor blocker for the FGF receptor, we then now get more cell death than we would have had with just the MCL1. All right, so I'm just going to kind of summarize these things. I think we're decent on time here. I've got a few more slides, and we should have some time for questions. So what we initially found with our collaboration with Ryan's group was these host-derived lung factors played a critical role in signaling through receptor tyrosine kinases and promoting ERK signaling and these downstream targets like MCL1 that promote cell survival. And we could target these with inhibitors. But what I've just showed you is that in the process of targeting this, we create a situation where the cells that we're trying to kill actually promote the survival of the cells next to them in somewhat of a random process. But it's not just one factor alone. It's the combination of this lysis-derived growth factor and the host factors that promote resistance of individual cells or persistence. I'm a little hesitant to call it resistance because we only measure it for a short period of time, so we don't know what would have happened 6 months from now, right? So they persist in the time period that we measure them, but we can't measure longitudinally, right? So, but I think this is still incredibly valuable because it allows us to try and target those cells, and then we can look in preclinical models. So, so this in total leads to these heterogeneous responses of individual cells, and we can measure that. Okay, so now how do we start to translate this into an actual finding that we can move to the clinic? So we started to ask the question of whether or not we could combine MCL1 inhibitors with RTK inhibitors to then more effectively treat these resistant or persistent populations. And so we thought about this a bit, and we conferred with Ryan's group a bit, and we've been thinking about this problem with our ongoing collaborations. And so we tested a number of different combinations. And the reason for this is that while we showed in the experiments that I just presented that these FGFR inhibitors do block some of this process, and they are important for increasing the response to MCL1 inhibitor, not every patient's going to be the same, right? And things may change over time. So some of these inhibitors that may be a little bit more broad in terms of targeting some of the other receptors and growth factor pairings that we saw in these lysates as the tumor lyses, we felt would be perhaps a bit more, or worth more consideration and more testing. And so that's what we did in this assay that I'm showing you, where we're just measuring responses to MCL1 inhibitor alone, or these different inhibitors and their combination with MCL1 inhibitor. And what we find is that FGFR inhibitors actually work, and I'll show you synergistically with MCL1 inhibitors, and these other inhibitors that are more broadly specific work at least as well, if not better, in that scenario. So we're still having the intended effect, but we may actually be having a better response by adding in a little bit of breadth into this. And so in doing additional exploration of these combinations, I'm just going to show you one, and this is actually just for the FGF receptor inhibitor, just to show you some relationships between different cell lines, because I don't want to show you just one thing. And what we find are some relative differences that really play into the thought process of using these broad specificity RTK inhibitors. So if we look at OS17 cells and we treat with a matrix of increasing MCL1 inhibitor or FGFR inhibitor, we can see a synergy relationship occur. So you get more killing than just additive relationships if you combine these two together. And so this number means that 95% of the cells have been killed at this concentration. So the more red, the better. And so we can combine these together to increase effectiveness. And this is true of also the OS186 cell line, where the combination is better than alone for either one. But then we start to get slightly more complicated relationships where, for example, the OS52 cell line, which is known to have a P10 mutation, which would promote increased AKT activity. And remember, I talked about ERK and AKT activity. We start to see where this relationship breaks down. And this is probably because these cells are starting to care less about their microenvironment because they have internal changes that continue to signal through these pathways, right? So they don't really care quite as much about the growth factors. And then if you move all the way over to a 143B cell line, which has RAS mutations that hyperactivate ERK signaling and presumably AKT signaling, you now have a relationship where they really don't care about their microenvironment. The FGFR inhibitor, has really no effect, and the MCL1 inhibitor actually has no effect as well, because this is very, very similar to if we had spiked in FGF2 or some other growth factor into the media, right? Because they're just constantly signaling they don't care about whether they got it from the environment. So this tells us a little bit about why we're going to have to think about how we apply these combined treatments based on what particular patient needs might be. And so we went back and tested this combination in the lung model. So this is just to get you a little closer to an in vivo setting, just to show you how these drugs work together in combination. So here's, in this case, I'm just showing the nucleus of the cells just for sort of clarity to be able to track individual cells. So in the control cells, we don't see any cell death or very little cell death over time. If you treat with MCL1 inhibitor, you see some cell death, but there are these persisting cells. So the arrows kind of help point to that. Whereas if you combine FGFR inhibitor and MCL1 inhibitor, you actually see quite a bit of cell death. And I don't know why this movie's not working. It's being funny. But this is just to demonstrate in the lung microenvironment that we're seeing that same synergistic effect of combining these drugs together. All right. So I'm going to just put this together and then have time for a few questions. So what I showed you using our lung model was that we found that there was this drug-responsive population that had low ERK activity in FRA1, which is in the tumor core. There's a partially responsive fraction that sort of sits on the periphery and dies a bit later. And then there's this resistant or persistent population that exhibits a rise in ERK activity and downstream ERK targets during the treatment period, and these guys persist longer. And so this is just showing you spatially what that looks like, where we have this sort of responsive, partially responsive relationship based on location. And what we find is that this relationship with persistence correlates with the cell lysis of the tumor core and release of growth factors into the microenvironment that activates ERK signaling and this sort of late onset and resistance phenotype. But if we co-target with MCL1 inhibitors and try to block this lysis-derived signal, we can actually enhance killing. The side benefit of this is that MCL1 inhibitors in the clinic have been, there's been some concern that at effective doses, there might be some on-target toxicity effects. However, we think that by combining these drugs together, we might be able to actually reduce the required dose of MCL1 inhibitor, and this is something that we're exploring, right? So trying to think about how we can maximize response while minimizing toxicity. And so just another note about, this is the last slide, is just to say that we have to think about what individuals need. We can't do we all know this, but I'm just stressing this again. We find that there are situations where there's combinatorial synergy between these drugs, but then some patients aren't going to respond. And in this case, the OS052 cell line leads us to believe that this, if this were a patient, they would respond to the MCL1 inhibitor, but the FGF inhibitor actually might hinder things and not have a benefit, right? Whereas other cell line models like the hyperactivated RAS, which thankfully is rare, this may not be effective at all. And that's because they're basically not caring about their microenvironment or what's released. They're just persistently signaling through these survivals that promote, or these pathways that promote survival. And then the last point related to that is that we really need to consider these broad specificity inhibitors, so long as they don't create a downside in toxicity, to really encompass the variation that may occur in these different pathways of interest that promote survival. And so with that, I think we're pretty good on time. I will take questions, and I just want to acknowledge again, Rowan, Jeremy, Vaibhav, and Carol, who have really been instrumental in getting this work to where you see it.
Thanks so much, Dr. Davies. That was great. And I think we already have a bunch of questions that came in. Walker, if you want to tackle those.
Yeah. I'll just start with the first question that came in. They ask, how do you think your data informed the interpretation of data reported by others? Initially, Alejandro Sweet Cordero, showing that FRA1, parentheses, the FOSL1 gene product, may be a protein that defines and reinforces a particular osteosarcoma phenotype. Some of the work he's presented suggests that tumors can change phenotype over time. Do you think that RUNX2 phenotype tumor cells will change into the FOSL1 phenotype tumor cells to traverse the bottleneck associated with early metastases?
Yeah, that's a great question. I'm waiting for this paper to come out, and I don't believe it's come out yet. I've talked to Alejandro a little bit about this. So it's a great question. You may see that actually some of that stratification where I showed the drug synergy may break into those that sort of dichotomy where it's the FOSL1 high is the sensitive, right? Because that's potentially an ERK-driven tumor, or has some intrinsic, FOSL1 high behavior. I think I can't answer the question because we haven't done the experiments to do it. I think it's something that we need to do and that I'm very interested in. The one thing that I'm not sure about in that regard is that presumably, not having read the whole paper, so I'm sort of speculating a little bit. Presumably, some of this was it was done in patients, and it was also done in cell lines. And so what's hard for me to know is that if those phenotypes are persistent in the absence or presence of exogenous growth factors. And so if I knew that, I would be able to answer the question a little bit more. Certainly, I think it's an interesting one, and I do believe that most likely the environment and the growth factors that are around could push in one direction or the other. So I don't know that they're necessarily fixed states. I'll leave it at that. I think I'm hedging a little bit, but I'm trying to answer the best I can.
Gotcha. No, like that's super confusing that it can change. So like I don't know. That's
presumably there's some intrinsic stability, but certainly, environment's going to matter. That's my argument all along with this, but also, treatment related changes in state may be a factor as well, right? And I just don't know the answer.
Okay. Thank you. I'll move on to the second question. So, the second question asks, do you ever see cells that appear to be stuck between states where phenotypic or activity-related markers are expressed for two different cellular states at the same time? If yes, are cells within the same spectrum of the heterogeneity you describe more prone to metastases, or do they appear to be more quiescent?
Yeah. So we haven't measured the state transitions in the way that this person's asking. So this is a strength weakness kind of situation with the model, is that we can only measure so many things because we can only put in so many different reporters, and so I would say, based on things like division and quiescence, I think we're seeing evidence that some of these persisters, for example, are entering into more of a quiescent type state, but we're observing them indirectly. So we're not measuring, say, a quiescence marker per se, we're measuring that they aren't dividing. So what we're in the process of doing is rather than sort of making that assumption that they are quiescent, is that we're now building into these models where we do the dynamic study where we're filming and measuring responses, but then at the end of the study, we're performing cyclic IF in these three-dimensional tissues to identify specific markers of quiescence. So the short answer is there's evidence of them entering different states like quiescence, or even in some cases like an epithelial mesenchymal-like state, like transitioning into, even though they're mesenchymal already, more of an invasive EMT-like state to try and get out of that environment as they're dying. But we're actively trying to develop methods to measure those specifically.
Gotcha. Okay, thank you. Well, in the spirit of time, I'll move on to the third. This is the last question that came in the Q&A. They ask, if you apply cyclic stretching of the lung tissue, do you think that would affect integrin-mediated outside and in signaling? And then I can let you answer that. And then there's like a second question after that they ask.
Yeah, no, that's an awesome question. There have been studies that show that like you can phenotypically revert cells if you squeeze them at certain times, right? Like you can take a tumor cell and squeeze it, and it'll form a normal structure or normal appearing structure, even though it's genetically mutant. So it's a great question. So we haven't done the cyclic stretching, and that like technically is really the challenge for us to be able to do that. That being said, you would be amazed by the forces and squeezing that you're seeing from just different interactions within the tumor environment. And there are other cell types moving around here, like immune cells that are deforming the matrix and stretching it while we're imaging. So still not another unsatisfactory answer, but I don't know the answer. I think it's a great question that we can't really test in the current form. I do see evidence that there is stretching and squeezing that doesn't seem to have a particular effect on the tumor cells, but that's purely speculation at this point. So I guess I'll take the second part of that question and hopefully I can answer it better.
Okay. So the second part just asks, did you apply fluorescent viability dye to the lung tissue to confirm host cells are alive?
Yes. Yeah, so okay. I didn't show that. What we've done is we've done live dead stains and mitochondrial activity assays to show that these things are still alive. And so we still see persistent living signals up to a couple of weeks. You do see some short-lived immune cell populations that are kind of residing in there. They will die, like a neutrophil will die and things like that. But the structural cells and the resident cells of the lung environment are pretty stable. And so the precision cut lung method that we generally use is fairly well established. That being said, we're pretty conservative in terms of wanting to film in the earlier stages as opposed to reaching out too far with this assay. Like we can culture them for a couple of weeks, but I start to question whether or not that's truly representative of the biology at that point.
Gotcha. Thank you. I think Christina may have a question or two.
Yeah, so first of all, can I say that this seems like 20 years of work or something in those slides? It was a lot of work as you kept layering on. And also like probably like 10 or 20 years ago, not possible at all, right? To be able to, that's like a tremendous amount of work. But I did, as you were going, had the sense of like, oh my gosh, I feel like it's one of those like the more you know, the less you know kind of dynamics, right, as we just continue to understand how complex the biology is. And so couple questions for you related, focusing more on like clinical translation. So really interesting how you kind of mentioned how this can direct in, clinically, like choosing a TKI. That comes up a lot all the time, and interesting kind of we have to be mindful of how kind of one thing can impact X, but then that also results in impacting Y. And so you showed 4 cell lines. Are you planning on testing some more cell lines so we have kind of that more comprehensive, as much as we can, of like all the different snowflakes in osteo and what that might look like?
So we have, the way we're approaching that is that, so short answer, yes. What we're trying to do is create a panel of responsiveness to growth factor, and a panel of responsiveness to the RTK inhibitors, and also to benchmark those against against drugs that are used, like, for example, ifosfamide or doxorubicin and those types of things that we know in a patient actually produce a response, or regorafenib, or some of the other things that have been shown in studies to produce a response. And so we're sort of trying to build out a catalog of those things and look at responses in a dynamic way that sort of map back and forth, so we can get some benchmarks of things that did work, things that didn't work, but also a greater picture of the spectrum of responses to different growth factors and variation from patient to patient, to the extent that we can measure that, right? But I think our system gives a really unique way of doing that, right? Because we can quantitatively measure that, how long, how much magnitude, or what is the magnitude of that change, in a very quantitative way. So that's our intent, is sort of build that catalog and put it out in the community so that people can use it as they see fit.
Yeah, and to me also, just a sense of timing seems really important. I mean, just because of the switching between the different phenotypes. And I'm also curious, just as we think about like clinical application, because oftentimes patients may have been pre-treated, depending on, at some point when they hopefully will have really great risk stratification, and we can kind of start applying these combinations earlier in a patient's treatment plan. But how can previous treatment impact these dynamics in the TME and in the tumor? So like if they've been treated with MAP or IFOS, like how does that start to change kind of what you're seeing?
Right. I think that's a really important consideration, because for a number of reasons. One, that changes the molecular landscape of the tumor itself, but that it has effects on the environment itself, the extracellular matrix, and all of these things, right? So then these cells regrow into a new environment, or a new environment from what they initially established themselves in, right? And so the question about the lung moving and the forces that puts on that. Well, the stiffness of the ECM and those types of things are going to have effects on it. So I think we need to be creative in our assay to find a way to do that. And I think we have that potential because you could imagine we can inject these reporters, treat the mice, for example, and let some of that die off, right? Some of the tumor cells die off and then regrow, and then we can test, untreated versus prior treatment, and ask whether there are differences in these responses. And then, I can't, stress enough that we have to pair this with patient data and really, what's coming out of the patients too. But I think we could use our model to ask some of those questions, right? And model the treatment that the patients are really seeing, and then what that pre-treatment effect would be. So I hadn't really thought about that, but I think that's a great suggestion.
I know we have to wrap up because we're already a little bit over, but I also just wanted to ask real quick, the MCL1 inhibitor, is that something that has been used clinically in humans? You mentioned that there were some like on-target toxicity issues.
Yeah, so the initial studies were done with an AstraZeneca compound, and there were some concerns about cardiac enzyme or troponin elevation. And so those studies were halted in phase 1 because of that, and I think in part because of some lack of clinical efficacy, because most of these drugs are developed around hematological malignancies and trying to treat those, and I think there's been a marginal effect. That being said, there are some newer generation inhibitors that give us some hope that this could be something that works, and so there's some newer data saying that they're more tolerable, or some of these drugs are more tolerable. Specifically, there's an Amgen drug that the toxicity, the signals are shown to be basically reversible and asymptomatic, which is a good thing. And then Gilead has an ongoing study, which is actually, there's a study here at OHSU for solid tumors. And that study is the only one that I know of that's in solid tumors. It's phase 1. And sarcomas are in the inclusion criteria, not specifically osteo, but it will be interesting to see what comes out of that. And actually, both Ryan and I are negotiating to get that drug to test it specifically in these models. So that's our hope. We're optimistic, but I think our preliminary data argue that we really need to see this through and to decide based on the evidence whether or not it's worthwhile or not.
Awesome. Well, thank you. We do have to wrap up, and thank you. Dr. Davies has generously agreed to share his slides, so we'll make sure you guys get a copy of that. And of course, he will be at Factor, so great place to kind of follow up with kind of more questions in person. Thank you so much, Dr. Davies, for joining us on OsteoBites today and for making it better for osteosarcoma patients. More information on this and all OsteoBites can be found on our website at mibagents.org and on the MIB Agents YouTube channel and at your favorite podcast place. We are going to be back here in 2 weeks. We are going to be joined by Dr. Chris Thompson on February 6th. Dr. Thompson is from Ethos Discovery, and he is going to discuss the MIMIC trial. That's the minimally invasive metastasectomy in canines trial, which will assess the feasibility and tolerability of VATS for lung mets and eventually deliver this care as an outpatient procedure. And in addition to assessing the therapeutic benefit in canine patients, the study will also pave the way to conduct accelerated studies to identify new drugs to prevent lung mets in both human and canine osteosarcoma. More lung samples there. You can find our OsteoBites lineup for the next few months on our website. And if you have any ideas for future topics you'd like to hear about, please feel free to email us at events@mibagents.org. Thank you again, Dr. Davies and Walker, for spending an hour with us today. And thanks to all of you for taking time out of your busy days to spend an hour with us. We hope to see you back here in a couple weeks on Feb 6th when we chat with Dr. Chris Thompson. Thank you, everyone. Thanks, Dr. Davies. Thanks, Walker.